We've all had a scraped knee, right? But have you ever really stopped to think about it? To follow that simple injury all the way down to the level of our DNA? Well, that's exactly what we're going to do. We're going to connect a common, everyday scab to the absolute cutting edge of genetic medicine.
Okay, so just picture this. Maybe it's a memory from when you were a kid, or hey, maybe it happened last week. You're riding your bike, you take a little spill, and bam, you land hard on the pavement.
You look down and, yup, there it is. A fresh scrape. A little bit of blood starts to well up. It's a totally normal, everyday kind of injury.
And just like you'd expect, after a few minutes, the bleeding slows down, and then it stops completely. Over the next day or so, a nice protective scab forms over it. We barely even think twice about it.
But have you ever stopped to wonder how our bodies actually pull this off? It seems so automatic, But underneath the surface, it is an incredibly complex and, honestly, kind of beautiful process.
That process is called the clotting cascade. And if you could zoom way, way in, down to the molecular level, you'd see it's less like a simple patch-up job and a lot more like a perfectly choreographed ballet. So, how many dancers are in this molecular ballet? Not one, not two, but 13. That's right. Your blood has 13 different protein molecules known as clotting factors, and they're all just waiting for the signal to act. And here's the cool part, how they all work together. When you get injured, a signal goes out. This activates the first clotting factor, which then activates the next, and so on and so on. It's this perfect domino effect, a chain reaction that builds and builds until it results in a solid clot, what you and I just call a scab. But that perfect chain reaction is also its biggest weakness. What happens if just one of those 13 dominoes is taken out of the line? What if a single one of those crucial proteins is just missing? Well, that is precisely what happens in a disease called hemophilia. The entire cascade fails. The chain is broken, a clot just can't form, and what should be a minor injury can lead to dangerous, prolonged bleeding. All because one protein is absent from the ballet. So why is that protein missing in the first place? To find the answer, we have to look past the protein itself and go even deeper, to the original blueprint in our cells. And that blueprint is our genes. The simplest way to think about a gene is like a recipe. It's a specific set of instructions that your cells read to know exactly how to build a specific protein, like one of our 13 clotting factors. And this right here is the root of the problem. In a healthy person, the gene has the correct, clear instructions. But in a person with hemophilia, that gene is faulty. The recipe is wrong. The instructions are either garbled or missing entirely. So the cell simply can't build the correct protein. So it's a really clear biological pathway. Our genes provide the instructions. Those instructions are used to build proteins, and those proteins determine our physical traits. Everything from our eye color to, yes, the ability to form a scab. So if the problem is a missing protein, the solution for many years has been remarkably direct. How do you help someone whose body can't make one of the essential clotting factors? You just give it to them. The primary treatment for hemophilia is to inject the missing clotting factor protein right into the patient's bloodstream. This provides the missing link in the chain, allowing their blood to clot the way it's supposed to. But this isn't a permanent fix. It's a constant, ongoing treatment. The body naturally uses up these proteins over time, so patients need these injections throughout their entire lives to make sure their blood can always form clots when they need it to. What's absolutely incredible is how we get this protein now. In the past, it had to be painstakingly separated from massive amounts of donated human blood. Today, thanks to biotechnology, scientists can take the human gene for that protein and insert it into hamster cells in a lab. I know, it sounds wild, but it turns those cells into tiny, efficient factories that produce the exact protein needed for treatment. But replacing the missing piece is still just managing the symptoms. So what if, instead of adding the protein, we could fix the original faulty blueprint so the body could just make its own? Well, this is the future we're all heading towards. This is the revolutionary promise of gene therapy. It's a potential cure that aims to go right to the source of the problem. By fixing or replacing a patient's faulty gene, their own cells could finally get the correct instructions and start producing the missing clotting factor all on their own. Think of it like this. Current treatments are like constantly adding water to a leaky bucket. You have to do it forever. Gene therapy is about patching the hole in the bucket itself. One addresses the symptom, the other actually fixes the source of the problem. Now, let's be clear, this isn't science fiction, but it's not quite standard practice yet either. Gene therapy for hemophilia is still being actively researched and developed, but it truly represents the next frontier in how we think about and cure genetic diseases.
And that leaves us with a final, really big thought. We've journeyed from a simple scrape on the knee all the way to the complex code of our DNA. We've seen how one faulty gene can cause a disease and how correcting it could offer a permanent cure. Which really makes you wonder, doesn't it? If we can do this for one, could fixing a single gene one day cure a world of diseases?